Welcome to our deep dive into the world of protein folding. We'll be looking at some recent Nobel Prize news and a Wikipedia article to understand how these tiny structures, essential for life, fold into complex 3D shapes. We're going to go from, basic science to the cutting edge of AI.
Yeah, it's amazing how these proteins, they're like the workhorses of our bodies, and they totally rely on being able to fold correctly. It's almost like a tiny origami project,
Yeah.
The final shape determines exactly what they do.
That's a great analogy, and our sources actually talk about this idea. Back in 1972, Christian Anfinsen won the Nobel Prize in chemistry for his work, showing that a protein's amino acid sequence basically dictates its shape, which then influences its function.
Yeah, Anfinsen's discovery was huge. It was like the first clue to solving this whole protein folding puzzle. Scientists realized that, at least in theory, they could figure out a protein's structure just by knowing the order of its amino acids.
So if we know the building blocks, why was it such a challenge to figure out the final folded product? Seems like it should be pretty straightforward, right?
Not exactly. See, a protein can fold in a crazy number of ways. We're talking like a mind-boggling number of potential conformations. Just to give you an idea, for a small protein, say, with only 100 amino acids, the possible number of folds is actually more than the number of atoms in the entire observable universe. This whole complexity is what's known as Levinthal's paradox.
Wow. That really puts things into perspective. So proteins don't just randomly try every shape until they find the right one, huh?
Nope, not at all. There are definitely guided pathways and steps along the way, but figuring those out has taken, decades of research. And it's not just some theoretical thing either. Misfolded proteins can clump together and cause all sorts of diseases, things like Alzheimer's, cystic fibrosis, even diabetes.
Okay, so the stakes are high. But then AI comes into the picture. I'm talking about AlphaFold, developed by DeepMind. Those are the same people behind that amazing chess-playing AI AlphaZero, right?
Exactly. AlphaFold is a perfect example of how AI is tackling these seemingly impossible problems in science. Basically, it was trained on this huge database of known protein structures and uses that knowledge to predict the shapes of new proteins with incredible accuracy.
So it's like AlphaFold learned the rules of protein folding from this giant library of examples.
Exactly. And it did such a good job that the 2024 Nobel Prize in Chemistry was actually awarded to David Baker, John Jumper, and Demis Hassabis for their work on protein structure and design, including developing AI to predict these structures. It's going to have a huge impact on things like developing new drugs and materials.
That's amazing. And speaking of David Baker, he also created a program called Rosetta, which lets scientists design brand new proteins from scratch.
Right. And Baker's team even developed this method called family-wide hallucination, which uses AI to imagine new protein combinations that don't even exist in nature. Think of it like giving AI a set of building blocks and letting it just dream up new, never-before-seen structures.
It's like we've cracked the code of life and can now write our own instructions. What are some examples of these new proteins that they've designed?
Well, one really cool example is a light-emitting enzyme. Basically, it's a protein that glows. And the possibilities for something like that are, they're endless.
That's mind-blowing. But AlphaFold doesn't stop there. It keeps getting better. What about AlphaFold3?
Right. AlphaFold3 is even more powerful. Not only can it predict protein shapes, but it can also pinpoint those potential binding sites for drugs. It's like giving researchers a treasure map to find the best ways to target specific proteins with, drug therapies.
And with DeepMind's Isomorphic Labs, which is a collaboration with all these pharmaceutical companies, they're actually turning those AI predictions into real treatments. It's a total revolution in protein design, and we're really just starting to understand the possibilities.
AlphaFold3 being able to pinpoint those binding sites is like a total game changer for drug discovery. It's like having this molecular magnifying glass, We can zoom in on the exact spots where those drugs interact with the proteins.
And having that level of precision has to be like speeding up the development of new therapies. Right. It's not just trial and error anymore.
Oh, absolutely. It's streamlining the whole process, helping researchers design drugs that are way more targeted and way more effective. And it's not just for diseases we already know about either. Think about tackling future threats, new viruses or those antibiotic-resistant bacteria.
So we're not just playing catch up, but we're actually getting ahead of the curve.
Yeah.
But I imagine there are challenges too. The sheer amount of data that AlphaFold generates must be just overwhelming. How do researchers even begin to sort through all of it?
That's a good point. The database is definitely huge, but it's also super well organized and easy to access. Think of it like a really advanced search engine for protein structures. Researchers can search for specific proteins, see what they do, even compare them to others.
So it's like Google, but for proteins.
Mm.
But it's not just about finding the information, right? This knowledge has to be like turned into real-world applications.
Exactly. That's where it gets really interesting. We've talked about drug development, but the potential uses for protein design go way beyond just medicine.
Like what? Give us some examples.
Well, imagine engineering proteins that can, like, break down pollutants in the environment. Cleaning up oil spills or detoxifying contaminated water.
Wow. So we could use protein design to, like, fight pollution. That's incredible. What else?
Well, there's agriculture. Imagine designing crops that are resistant to, say, pests or diseases, so we don't need to use as many harmful pesticides. Or even crops that can grow in tough conditions, like drought or extreme temperatures.
That would be huge for food security, especially with climate change being such a big problem.
Exactly. And don't forget about material science. We could design proteins to create biodegradable plastics or even new types of building materials that are stronger and more sustainable.
So we're talking about a future where protein design could revolutionize everything. Healthcare, environmental protection, even how we build cities.
It really is a future full of amazing possibilities. But of course, with great power comes great responsibility, right? We need to make sure that all of these technologies are developed and used ethically and carefully, thinking about how they could impact society and the environment.
Yeah, that's a really important point. We can't just rush ahead without thinking about the consequences.
Definitely. For example, as we start being able to create proteins with totally new functions, we really need to think about the risks of, like, releasing them into the environment. Could they disrupt ecosystems or even have unintended consequences? These are questions we need to be asking ourselves.
And it's not just the environment. There are social impacts, too. What about access to these technologies? We need to ensure that the benefits of protein design are shared equally and don't just make existing inequalities worse.
You're absolutely right. These are really complex issues that need careful thought and open discussion. We need scientists, policymakers, ethicists, and even the public all talking to each other.
Okay, so we've got this incredible potential, but we also need to be cautious and thoughtful. It's a fascinating but pretty challenging landscape to navigate. But let's get back to the science for a minute. We mentioned earlier that AlphaFold has already predicted the structures of, like, over 200 million proteins. What's that actually mean for researchers? How are they using this information?
Well, this database of protein structures is basically a goldmine for researchers. It's accelerating research in so many different fields. For example, take the study of diseases. By understanding the structure of the proteins involved in a specific disease, researchers can figure out what to target with drugs.
So it's like having a blueprint, showing them exactly where to aim their therapies.
Yeah, that's a great way to put it. And it can also give us a much deeper understanding of what causes those diseases in the first place, which can help with developing new diagnostic tools and, preventive measures.
So it's not just about finding cures, but it's about understanding the root of these illnesses. But I'd imagine this knowledge also goes beyond human health, right?
Definitely. For instance, in bioremediation, researchers are looking at using proteins to break down those pollutants we were talking about earlier. By knowing the structure of those proteins, they can engineer them to be even better at cleaning up those contaminants.
So we're using protein design to fight pollution on a molecular level. That's amazing. What about applications in, like, industry?
Oh, there are tons of possibilities there too. Enzymes, which are a type of protein, are already used in lots of industrial processes, from food production to making textiles. By knowing their structures, we can engineer them to be more efficient and work under very specific conditions. That can lead to processes that are more sustainable and cost-effective.
It's like we're giving nature a helping hand, optimizing these biological machines to work even better. But we're not just tweaking existing proteins, are we? We're also entering this whole new territory of designing entirely new proteins with functions we've never seen before in nature.
That's right. And that's where things get really exciting. We're going beyond just copying nature. We're becoming like architects of the molecular world. Who knows what incredible things we'll be able to do as we explore this new frontier.
Okay, so we've talked about the huge potential of protein design and the responsibility that comes with it. We've seen how this knowledge is being used in all these different fields: medicine, environmental science, industry. But before we wrap up, I want to talk about something that's both fascinating and a little bit unsettling: protein misfolding.
Ah, yes. the dark side of protein folding. It reminds us that this delicate dance of molecules can sometimes go wrong, and that can have some serious consequences.
Yeah, right. We talked earlier about how misfolded proteins are involved in diseases like Alzheimer's and Parkinson's, but let's dig a little deeper. What exactly happens when a protein misfolds, and why is it so bad for our health?
Okay, so imagine a protein like a carefully folded origami crane, right? When it's folded the right way, it's beautiful and it works perfectly. But if you mess up the folds, it becomes just a jumbled mess.
Yeah, that's a great analogy. So when a protein misfolds, it's like that messed up origami crane. It loses its shape and can't do its job, right?
Exactly. And not only that, but these misfolded proteins, they can be kind of sticky. They tend to clump together and form these aggregates, and those can really mess up normal cellular processes.
It's like they're gumming up the works of the cell.
Exactly. And these aggregates can actually be toxic.
Yeah.
They can lead to cell death and damage to tissues. And this is what happens in diseases like Alzheimer's, where you get those amyloid plaques, which are made up of these misfolded proteins building up in the brain.
So it's not just that the misfolded proteins aren't working right, they're actively causing harm. But why do proteins misfold in the first place? Is it just random, or are there specific things that trigger it?
Well, it's kind of a mix of both. Sometimes errors happen during the folding process itself, especially when there's stress like high temperatures or changes in pH. And also certain mutations in a protein's gene can make it more likely to misfold.
So it's like a combination of external stresses and internal vulnerabilities that can cause these misfolding events. But what's really interesting and kind of scary is that these misfolded proteins can sometimes set off a chain reaction and cause other proteins to misfold too.
Yeah, that's the idea behind prions, which are these misfolded proteins that can actually make other normal proteins misfold as well. They're like a bad influence, spreading that misfolded shape to their neighbors.
It's like a domino effect of protein misfolding, and this prion thing is involved in some really serious neurodegenerative diseases, isn't it?
That's right. Diseases like Creutzfeldt-Jakob disease, CJD, and bovine spongiform encephalopathy, BSE, or what we call mad cow disease, are caused by prions.
So we've got this whole complicated mix of factors that contribute to protein misfolding, things like environmental stresses, genetic predispositions, and those prions. But what are researchers doing to fight this problem? Is there any hope for preventing or treating these misfolding diseases?
It's definitely a challenging area of research, but scientists are making progress on a few different fronts. One approach is to develop drugs that can help stabilize proteins in their correct shape and stop them from misfolding in the first place.
So it's like giving the proteins a little extra help to fold the right way.
Exactly. And another strategy is to target the processes in the cell that are responsible for protein quality control. Our cells already have these really complex systems for finding and getting rid of misfolded proteins, and researchers are looking for ways to make these natural defenses even stronger.
It's like giving the cell's cleanup crew a boost. But what about those sticky aggregates that have already formed? Is there a way to break those up?
That's a tough one, but scientists are exploring different options, like using antibodies or small molecules to target those aggregates and dissolve them.
It's like finding a special solvent that can break down those protein clumps. And we also mentioned earlier those chaperone proteins. Could those be used as a treatment?
Yeah, that's another area of active research. Chaperones are like protein folding assistants, and scientists are trying to figure out ways to increase the amount of them in cells or enhance their activity.
So it's like a multi-pronged attack on protein misfolding, trying to prevent it, strengthen the cell's defenses, and even break down those aggregates that are already there.
Yeah.
It sounds like a promising but really challenging field of research.
It definitely is, and all the advances in protein science, especially with AI, are giving us new tools and insights to tackle these challenges.
It's amazing to think how far we've come from trying to figure out the order of amino acids in a protein to being able to predict and design them with AI. It's incredible what humans can do.
And remember, this is just the beginning. The field of protein science is constantly evolving, and with every new discovery, we're understanding more about the very foundations of life.
So as we wrap up this deep dive into protein folding, what's the one big thing you want our listeners to think about?
I think the most important thing to remember is that proteins are absolutely essential for life, and whether they fold correctly is crucial for how they work. When protein folding goes wrong, it can cause all kinds of diseases, but with all the knowledge we're gaining and the advances in technology, we're starting to understand and even control this incredibly complex process, and that's opening up possibilities for new treatments and solutions to some of the biggest challenges facing humanity.
That's a great point. We've gone from the basics of protein structure to the cutting edge of AI, exploring both the amazing potential and the possible risks of this incredible field.
It's been a fascinating journey.
It really has. And with that, we come to the end of our deep dive into protein folding. Thanks for listening, everybody.